somewhere in a laboratory right now. A scientist is trying to build life, not life as we know it, not life that follows the 4-billion-year-old rules that every single organism on this planet obeys. Something different. Something that, if it were ever released into the world, could make every antibiotic useless, every immune system invisible, every ecosystem on Earth defenseless. Scientists call it mirror life. And in December 2024, 38 of the world's most respected biologists, including 2 Nobel Prize laureates, published a paper in one of the most prestigious scientific journals on the planet, saying the same thing. We need to stop, not slow down, not proceed with caution. Stop. One of the paper's authors, David Relman, a professor at Stanford University, who has spent his career investigating biological threats, including the 2001 anthrax letters and Havana syndrome, said something that stopped the scientific community cold. He called mirror life the first plausible existential risk he had ever encountered in a long career of studying ways the world could end. Not a nuclear threat, not a pandemic, not climate change, mirror life. This is what it is. This is why it terrifies them. And this is why the conversation happening quietly in laboratories and policy meetings right now may be one of the most important conversations of the 21st century. To understand mirror life, you first need to understand something fundamental about how all life on Earth is built. Every living thing on this planet, every bacterium, every plant, every animal, every human being, is built from molecules that have a specific handedness. Think about your hands for a moment. Your left hand and your right hand are mirror images of each other. They are the same shape, the same size, the same structure, but they are not identical. You cannot superimpose one on the other. A left-handed glove does not fit a right hand. Molecules work the same way. The amino acids that make up every protein in your body are left-handed, every one of them. The DNA and RNA that carry your genetic code are built from right-handed nucleotides. This is not a coincidence. This is not random. This molecular handedness, scientists call it chirality, is one of the most fundamental and universal features of life on Earth. It has been this way for over 4 billion years, and it matters enormously because the entire machinery of biology, enzymes, immune systems, cellular receptors, the mechanisms that allow cells to communicate, replicate, and survive, all of it was built around this chirality, evolved around it, optimized around it over billions of years of natural selection. Now imagine flipping it. Mirror life would be built from the opposite-handed molecules. Right-handed amino acids instead of left. Left-handed nucleotides instead of right. A complete molecular inversion of everything that natural life is made of. And here is why that is so deeply dangerous. All of the biological systems that have evolved to interact with natural life, immune systems that recognize pathogens, enzymes that break down cellular material, viruses that infect bacteria. They work by recognizing specific molecular shapes. Lock and key. The lock is built for one specific shape. The mirror image key does not fit. Mirror bacteria would be invisible. Not invisible like camouflage. Invisible like they don't exist. Your immune system would never see them. The antibiotics designed to disrupt bacterial cell processes would have nothing to work on. The bacteriophages, viruses that kill bacteria and are one of the primary checks on bacterial populations in every ecosystem on Earth, would be completely unable to infect them. A mirror bacterium would be, in the words of Nobel laureate Jack Szostak, 100% certain to be resistant to all normal phages. It would grow and have almost nothing capable of stopping it. Here is where the science becomes genuinely frightening. We are not talking about a theoretical future technology. The building blocks already exist. Scientists have already synthesized mirror DNA, mirror RNA, mirror enzymes, fully functional biological machines built from the opposite-handed molecules. These are not hypothetical. They exist in laboratories right now. The technology to assemble a complete mirror cell from these components does not yet exist. Most experts estimate we are at least a decade away from the capability to create a mirror bacterium from scratch. But George Church, Winthrop Professor of Genetics at Harvard Medical School, and one of the most prominent geneticists on the planet, disagrees. He says it is less than a year away, not 10 years, less than 1 year. From the research already published, from the techniques already developed, from the knowledge already in the open scientific literature, it's already gotten to the point, he said, where somebody who is sufficiently mischievous could start with what we've already published and run with it. The gap between where we are and where we need to be to create mirror life is narrowing. The question is not whether it is possible. The question is whether anyone will do it before the world has a chance to decide whether it should be done at all. And that is a question that, as of today, has no binding answer. Kate Adamala is a synthetic biologist at the University of Chicago and one of the 38 scientists who signed the December 2024 paper. She used to work toward creating mirror cells. She stopped. The reason she stopped is the reason the paper was written. And it is worth understanding precisely, because it is more disturbing than it first appears. The original scientific interest in mirror life was medical. Mirror molecules, specifically mirror peptides and mirror proteins, have a remarkable property. Because they are not recognized by the body's natural degradation systems, they last far longer in the bloodstream than their natural counterparts. This makes them potentially powerful tools for drug development, particularly for cancer therapies that need to remain active in the body for extended periods. There was a real scientific logic to pursuing mirror biology for these reasons. But the more Adamala and her colleagues explored it, the more they began to understand something they had not fully appreciated. The properties that made mirror molecules potentially useful in medicine, their invisibility to biological recognition systems, their resistance to degradation, their inability to be detected and destroyed, were the exact same properties that would make a complete mirror organism catastrophic. You cannot have the useful properties without the dangerous ones. They are the same thing. Adamala describes the moment of realization. The team started trying to poke holes in the concern. They looked for ways in which the risk was overstated. They looked for natural checks, predators, immune responses, ecological limits that would contain a mirror organism if it were released. They found almost none. The more we looked, she said, the more we were certain. And more people were coming on board to this idea that there actually is no safe way to make a mirror cell. No safe way. That is not a precautionary statement. That is the conclusion of some of the best synthetic biologists on Earth after specifically trying to prove themselves wrong. So, what would actually happen if mirror bacteria entered the environment? The scientists who wrote the December 2024 paper modeled several scenarios. None of them are reassuring. Mirror bacteria would be capable of infecting every living thing on Earth that has a biology, which is every living thing on Earth: plants, animals, humans, other bacteria. because the immune systems of all of these organisms are built around recognizing left-handed molecular structures. A right-handed organism would be to all of them essentially invisible, not just to the human immune system, to every immune system on Earth. Every organism that has spent billions of years evolving defenses against biological threats has evolved those defenses against threats with a specific chirality. Mirror life has the opposite chirality. The defenses would not activate. Bacteriophages, the viruses that keep bacterial populations in check in every soil, every ocean, every ecosystem on Earth, would be useless against mirror bacteria. Bacteriophages work by injecting their genetic material into a bacterial cell and hijacking its replication machinery. The machinery of a mirror bacterium is inverted. The phage cannot interface with it. Mirror bacteria in the soil could displace natural bacteria. The microorganisms that make soil fertile, that cycle nitrogen, that underpin the food chains of every ecosystem on Earth. Mirror bacteria in the ocean could interact with the vast microbial communities that produce a significant fraction of the world's oxygen and form the base of marine food chains. And mirror bacteria, if established in the environment, could not be recalled. This is the key point, the one that makes this different from most biological risks. There is no cleanup operation for a self-replicating organism that is invisible to every natural predator and immune system on Earth. Once it is out, it is out. The consequences would unfold on biological timescales, which means they could compound for years, decades, or longer before the full extent of the damage was even visible. The authors of the science paper describe this as an unprecedented risk, not just in the sense of being serious, in the literal sense of being without precedent. There is no historical analog for what happens when a self-replicating organism enters the biosphere that nothing in the biosphere can detect, attack, or control. The response from the scientific community has been significant. And it has also revealed a genuine divide. The 38 scientists who signed the December 2024 paper are calling for research into mirror organisms, specifically the creation of self-replicating mirror systems, to be paused. They have proposed international discussions among scientists, governments, funding agencies, and policymakers to establish limits on where this research should be allowed to go. In February 2025, nearly 100 researchers, funders, and policymakers signed a statement arguing that mirror life should not be created unless future research convincingly demonstrates that it would not pose severe risks. A nonprofit called the Mirror Biology Dialogues Fund has sponsored a series of meetings specifically to develop recommendations. Scientists met in Manchester in September 2025 to discuss where the red lines should be drawn. But not everyone agrees that the alarm is warranted at this stage. Andrew Ellington, a molecular biologist at the University of Texas at Austin who develops synthetic organisms, pushes back. He argues that a mirror bacterium would be at a competitive disadvantage in natural environments, that the very features that make it invisible to immune systems would also make it unable to access the molecular machinery it needs to thrive. He has compared the early warning to banning the transistor because you are worried about cybercrime 30 years in the future. His concern is different, that premature regulation will stifle beneficial research in mirror biology that could produce real therapeutic benefits for real patients with cancer and other serious diseases today. This is not a simple debate between alarmists and dismissers. It is a genuine scientific disagreement between serious people about the magnitude of a risk that by definition does not yet fully exist. What makes it uniquely difficult is the asymmetry of outcomes. If the alarmists are wrong, and mirror life turns out to be less dangerous than the December 2024 paper suggests, the cost of caution is some delayed research and some missed therapeutic opportunities. If the alarmists are right, and mirror life is released into an environment that cannot contain it, the cost of not being cautious enough is something for which there is no precedent and no recovery. There is a question underneath all of this that the scientists who wrote the December 2024 paper are explicitly trying to raise. Not just with other scientists, with governments, with the public, with anyone who will listen. It is this: should the fact that something can be done determine whether it will be done? The history of science contains several examples of moments when humanity gained the power to do something before it had fully reckoned with whether it should. Nuclear fission, gain-of-function research. The early days of recombinant DNA technology, when scientists themselves called for a moratorium on their own work until the risks could be assessed. That moratorium happened in 1975 at the Asilomar Conference in California. Scientists paused their own research, met, debated, and established guidelines that governed the field for decades. Many regard it as one of the most responsible moments in the history of molecular biology. The authors of the December 2024 paper are explicitly invoking Asilomar. They are calling for the same kind of collective pause, the same kind of proactive reckoning with a risk that is not yet real, but is becoming more technically feasible with every passing year. David Relman, who described mirror life as the first plausible existential risk of his career, said something that captures the stakes with unusual clarity. He asked whether scientists could be the Dr. Ian Malcolm of Jurassic Park, the character who warns before the dinosaurs escape that the question is not whether you could, but whether you should. The dinosaurs in Jurassic Park were contained behind electric fences. Mirror life, once created, would have no fence. This is where we are. A technology that does not yet fully exist. A risk that cannot yet be measured precisely because the organism that poses it has not been created. A scientific community genuinely divided over the magnitude of the danger and the appropriate response. And a timeline that, depending on who you ask, runs from less than 1 year to several decades. What is not in dispute is this: the building blocks of mirror life exist in laboratories today. The knowledge required to advance toward a complete mirror organism is published and accessible, and the natural systems that have protected all life on Earth for 4 billion years, immune systems, predators, ecological checks, were not designed for this. They evolved in a world where all life had the same handedness, a world where that assumption held has existed for 4 billion years. The question now being asked, very quietly, in laboratories and conference rooms and policy discussions across the world, is how much longer that assumption will hold. This is Terrum, and this story is still being written.